Hello everyone, welcome to Mbbs Hub. In today's video, we are going to see about scientists and microscope. It is from microbiology. Okay, let's go into the topic. First one, Anthony Phillips van Leeuwenhoek. He first observed the bacteria and other microorganisms. He used a single lens microscope made by him, and then Louis Pasteur. He is the known as father of microbiology, and he proposed so many things and introduced. He proposed the principles of fermentation and introduced sterilization techniques, and described the method of pasteurization of milk. And he also innovated the theory of diseases and used nutrient broth to grow microorganisms. And then Joseph Lister, he is the father of antiseptic surgery. And then Robert Koch, he is invented the solid media for the culture of bacteria and pure culture and hanging drop tests about the mobility. And then he also discovered the bacteria. And we are listening about the coach postulates. And then Paul Ehrlich, he is the father of chemotherapy. Okay, and then Edward Jenner, he invented the vaccine for the smallpox. Okay, and then Ignatius Semmelweis, he told about the hand hygiene. And then Hans Christian Gram, he introduced about the gram staining. Okay, gram positive and gram negative, okay. And then Struska is the founder of electron microscope. And Alexander Fleming, he discovered the most commonly used antibiotic substance, penicillin. Alexander Fleming. And then Walter Gilbert and Frederick Sanger, these both developed the DNA sequencing. And Caris Bullis, he invented the polymerase chain reaction. Okay, discovered the polymerase chain reaction. And coming to the next topic, microscopy. In microscopy, there are five types of microscopes: bright field microscope, dark field microscope, phase contrast microscope, fluorescence microscope, and electron microscope. General microscope means what? In which we will see about the microscopes are the instruments in which we will see the microorganisms. Okay, in general, we observe the microorganisms and small cells. What are the microorganisms? Like bacteria, virus, parasites, and fungi. Generally, the bacteria are from 0.2 to 1.5 mm in diameter, in length. Okay, and in diameter, and 3 to 5 in length. The viruses are from 20 to 30, 20 to 300 nanometers in diameter, and these parasites are from few millimeter to several meters. And coming to the fungi, those are both two types: cylindrical and round or oval. The cylindrical are from 2 to 10 mm in diameter, and round or oval are from 4 to 8 mm in size. Coming to the microscope, it is a bright field microscope. These are ocular lens. These are resolving non nose piece. These are objective lens, 10x, 40x, 4, 10, 40, 100x. We will see now. We will also observe in the on our first year of Mbbs, and it is this is the stage. This is light source. From this, we all know about the microscope. And coming to the bright field microscope, coming to the properties of microscope, we will discuss about it. There are three properties of microscope. First one, there should be good resolution, good contrast, and good magnification. In good resolution, there should be refractive index. It is the main factor. The oil refractive index is more than the air refractive index. Okay, so the oil is used in for the RBC for identification. We will use not the RBC. Now first year we used oil. Okay, and for the gram staining also we will use oil, cedarwood oil, and there should be good contrast. uh, Good contrast means more visibleness, and by it will be improved by the staining, and good magnification. It will be improved by the convex lenses. It is there will be good magnification in the convex lenses. Coming to the bright field microscope. In bright field microscope there will be dark image with a bright background. Okay, the background will be white color. There will be bright. Okay, and the image will be dark. Okay, we will see organism in a dark color with a bright background. And coming to the dark field microscope, there will be a bright image. In this we can see there will be a bright image with a dark background. This is the background. This is the background, and this is the image. We will see it is a spiral coil bacteria. Okay, this is a dark field microscope. Coming to the phase contrast microscope, the contrast will be enhanced. In this we can see the cell wall is glowing, and we also observe the cells are contrasted. Okay, this is a phase contrast microscope. These are free living amoeba, Naegleria. Okay, and coming to the next microscope, fluorescence microscope. Fluorescence microscope. It uses the fluorescence property. The dyes will be exposed to UV, and the excited and the short wavelengths will convert into the long wavelengths. And the image will be formed. And okay, I will discuss about this. In bright field microscope, the light will be fallen on the condenser, and it will travels to the specimen. There will be both reflected and unreflected light will follows to the objective lens, and goes into the ocular lens. Then we will see. Okay, in the dark field microscope, we only reflected light will be passes through the objective lens. Okay, only reflected light will be passes through the objective lens, and then we will see through the ocular lens, but not the unreflected light. The unreflected light will pass through outside. Okay, in fluorescence microscope there will be applications, autofluorescence, and microscope. Microbes are coated with fluorescent dye. Okay, these are stained. Okay, stained non-specifically. The specimens will be stained in the here microbes. Okay, coming to the next microscope, electron microscope. electron microscope. In electron microscope, the electrons are a source of illumination. Okay, in this it is founded by the Ernst Ruska. Okay, in 1931, there is a transmission electron microscope. Transmission electron microscope. In this, the image will be very thin specimens. Okay, very thin specimens. There will be fixation. In this process are there. First one, there is a fixation, and then dehydration, and third one embedding, and then fourth slicing. In fixation, the cells are fixed. And these are killed. Okay, the cells are killed by using a glutaraldehyde. Glutaraldehyde. 1 minute. Or osmium tetroxide. For the stabilization. Okay, in dehydration, it is dehydrated with the organic solvents, acetone or ethanol. These are organic solvents. Okay, then embedding. In embedding, it is embedded in a plastic polymer. Okay, it is water insoluble. Plastic polymer is a water insoluble. Okay, and there will be complete dehydration will be done. If it is water insoluble, then dehydration will happen completely, and before embedding it should be completely dehydrated specimen, and it is cut into the thin slices. The slices will be very thin. This is the process for the to observe a microorganism in a transmission electron microscope. So these are the types, and in this there are in electron transmission electron microscope there are some applications like virus detection, contrast of EMI, and freeze contrast of electron microscope, and freeze etching technique. It's a alternate pathway for the specimen preparation. Alternate pathway. Okay, so this is our today's topic, guys. Thank you for watching.